Hello, today we will discuss about the fundamentals of bioinformatics. Actually, bioinformatics is a conglomeration of different streams, different disciplines like biology, computation, uh, information science, mathematics, statistics, all these things. Now, we'll see how this is useful and how this particular topic is growing very fast in these days. What is actually bioinformatics? It is an interdisciplinary field. It combines biology, computer science, statistics, mathematics, and information technology to develop different types of software tools for understanding biological data, biological models, the models, mathematical model developed from uh, analyzing those biological informations, and it also focuses on DNA and proteins sequences, because these are the fundamental units of life of that information. It carries the information which has been inherited from their parent to the offsprings. So, this DNA and protein sequence, they are very useful for understanding the behavior, for the characteristic of any organism. The key information, actually it is your equation like your bioinformatics is equals to its computation into biology, and the goal is to analyze, is to convert complex data uh, and analyze them, complex biological data or data which has been collected from the physiological processes in our body, and then we have to analyze those data using some software tools in computer. Then as we have already started discussing that this is a multidisciplinary uh, branch where the biologist will collect molecular data like DNA, then protein sequence, then gene expressions, all these things. Then collecting those data will be given rise to the data set is given to the computer scientist, and looking at those data or analyzing those data, computer scientists will develop tools, algorithms, softwares, and they will try to analyze the sequence, analyze the pattern which has been collected by the biologist. Then the mathematician will establish the statistical framework and the mathematical models, and finally the bioinformaticians, they will synthesize all disciplines, what has been collected and analyzed and modeled by the above three. Then basically it more or less it is just related to molecular biology. The fundamentals remain behind it. So that means it is all starts from that genetic information storage unit, that is your DNA, is your deoxyribonucleic acid. So that contains all the genetic informations in a double helix structure, and where those adenine will pair with thymine and cytosine will pair with guanine, and in this manner a human being contains 3 billion base pairs. We call these pairs base pairs, and their arrangement or their sequence will differ in approximately 3 billion different ways. Then once the DNA will be transformed or it will be transcribed to RNA. So RNA is like your ribonucleic acid. So whatever information stored in the DNA as genetic form, that will be transformed or into the, or we call that transcription. Through the process of transcription, it will be converted to RNA and act as a template for protein synthesis. What are the different types of proteins that will be synthesized in a particular human being for different activities, different physiological activities, that has been stored inside a DNA, and that through transcription process it will move to the RNA, and from RNA the protein will be synthesized through the process of translation. The protein will perform cellular function. We have in our body, we have different types of proteins, and all the building block of those proteins are amino acids. So 20 different amino acids are there, and their combination will give rise to multiple number of proteins. So through protein synthesis, this information is actually in a one-way traffic, never reverse. Means from DNA the transcription will occur and it will move to the RNA. From RNA the translation will occur and it will move to the protein. Then, as we know previously, DNA structure and genetic code, they are present, they are differ from person to person. This DNA double helix, they have 4 bases, or we call them base pairs, A with T and C with G. This base pairing always they follow A pair with T and C pairs with G, and they are connected, they are supported by a backbone of sugar phosphate in the direction of 5 to 3. So here you can mark this picture where clearly you can mark different base pairs, and in human basically we can have a 3 billion base pairs and 24 distinct chromosomes. We know that in our human body we have 23 pairs of chromosomes, but among them we have 24 distinct chromosomes. Now some terminologies we must know about that. Genome means organism's complete set of DNA. The complete set of DNA, complete hereditary information in a particular organism is known as genome. Gene is the discrete unit of that hereditary information, and specific DNA sequence encoding proteins. Means as a whole, all the different genes consisting or present inside a particular organism is known as genome. Individually we call them genes. Each gene, they will related to some protein. Every encoding sequence will give rise to a distinct protein. So chromosome is a structure containing genes. Human have 24 distinct chromosomes, as we have said previously. Genotype means genetic makeup of organism, that will be inherited information. Genotype. Every time we will have a genotype and we will have phenotype. Genotype means genetic makeup of organism, that will be inherited information from the parents to the offsprings. But phenotype is the physical expression of the trait, what we observe. And nucleotide at the basic building block of DNA, like adenine, thymine, cytosine, guanine, and in case of your RNA, the thymine is replaced by uracil. And just like analogy, if you can take, nucleus is just like library, and inside nucleus we have the chromosomes. As in a library, book shelves are there, and genes are like the books in the bookshelves. So protein synthesis it occurs from the genes to protein. How it occurs? Because we have different genetic code translations. Genetic code translation means 3 different nucleotides, we call that a codon. Each codon will give rise to each combination of these 3 different nucleotides will give rise to a particular or a specific amino acid. So, as you can see from the table, if we have all U, means we have all 3 uracils, then that amino acid will give rise to a phenylalanine. Then if it is UCU, then serine. Then UAU will give rise to tyrosine. UGU is cysteine. So like that, we have different codons which will give rise to different amino acids. So start codon is AUG, which is known as methionine, and stop codons are UAA, UAG. UAA means you have different UAG as and UGA. All these are 3 different stop codons. So small mutation in DNA can lead to amino acid change, and as amino acid changes, the protein synthesis also altered, and we are getting different proteins. Different proteins will give rise to diseases. So this is the fundamental behind this genetical disorders. So 3 sub-disciplines of bioinformatics are sequence analysis, structural analysis and functional analysis. So basically, as we have discussed, if this amino acid, this codon changes, then we are getting change in amino acid. As the amino acid changes, then there is a alternation in protein function, and disease will be created inside our body, or disease will be the result of that. So sequence analysis will give rise to DNA or protein sequence comparison, alignment algorithms, homology detection and gene identification. So these are the sub-disciplines where we can analyze the sequence of these codons, and the tools basically used are BLAST, then cluster IW. Similarly, we have structural analysis. Structural means 3D protein structure, because proteins they are actually 3D structure, 3-dimensional structures. They have different foldings. Those foldings can be predicted through different softwares. Similarly, we have RNA secondary structure, structure function relationship. So those structural analysis can be possible through tools like PyMol, Swiss model, all these things. And the third one is your functional analysis. Functional means how this gene will express themselves, how this protein-protein interaction will take place, then metabolic pathways that will give rise to your diseases. So we can understand, due to the change in this protein synthesis or in the codons, then or in the DNA gene expressions, we are getting the disease. That means the mechanism of disease can be better understood. So the tools we can use for analyzing the functions are KEGG and string. Now major bioinformatic database. So we have different databases already available and on internet. So they are very useful, like GenBank. They are actually the database of nucleotide sequence. They contains DNA and RNA sequence of number of persons, and it is public repository. UniProt is a protein sequence. Then PDB is a protein structure. They are given it to 3D structures. KEGG is pathways, metabolic pathways, molecular networks. Genoontology is gene functions, gene annotations and standardized terms. So all these databases, they are publicly available. We can also access them. Some of them can be accessible, and we can find different sequences like DNA sequence, protein sequence, molecular structure of proteins, and the metabolic pathways. All these things can be analyzed and understood from those database. Then database properties. Why? Because organized data storage, easy researcher access, because researchers are trying to combine or synthesize different types of proteins and their involvement in diseases. Annotation, cross-linking, curable. So these are some of the database properties. Database must be organized data, means it must be a structured data. It must be annotation, assigned functional characteristics, means we have to label them. Then cross-linking, there must be some information correlation and hierarchies in between them, those data. And queryable means rapid retrieval of specific information from those structured data or structure label data. So key bioinformatics tools, as we have seen, that is a BLAST. BLAST is basic local alignment search tools. It finds similar sequence in databases. It identifies the homologous gene and proteins critical for sequence comparison. It will identify, but critical for sequence comparison is not very easy in this tool. Then cluster IW is a multiple sequence alignment algorithm. They reveals evolutionary relationship used for phylogenetic analysis. And primer 3 is a design specific primer for PCR. They are essential for molecular experiments, and all these three are freely available for researchers. Then bioinformatic applications are vast. Molecular level, we can sequence analysis, structure prediction, protein structure prediction actually, then function annotation, mutation and identification. So that means if we have a database from of a means a generation to generation of a particular person, then how his behavior, how the gene expression, genotype and phenotype, they are differing, and we can also observe the difference and similarity among these current person and his previous or predecessors. So it can be defined, it can be analyzed, and how it is affected by the gene sequence, how the affected by those DNA sequence, it also be predicted. So then we have some system level applications like pathway analysis, protein interactions, gene regulatory networks, how this gene expression they are regulating the behavior from generation to generation. Then disease modeling. Disease modeling means observing those informations from different generations. We can model that disease, that due to this particular change in the sequence, this person in his previous generations, now this person is facing this disease or having this disorder. So it can be understood, we can model that. Then clinical applications like drug discovery, personalized drug or personalized medicine. Then we can have a gene therapy. If there is some defective gene, then we can also cut that. and disease diagnosis, everything can be done at a molecular level, and we can, if we can find what is the root cause at the molecular level, we can treat that problem at the molecular level, and it can be personalized also, and different person to person we can develop a personalized medicine. We can discover different types of, according to the change in the protein sequence or protein synthesis, we can also develop new drugs for treating those problems. Then biotechnology is one other use. Under biotechnology, we can also improve the crop, microbial application, antibiotic resistance, forensic analysis. All these things are coming under the applications of bioinformatics, very useful branch. Now, genome annotation is just like marking genome to identify regulatory sequence and protein coding regions. So, in that we have two parts, one is a promoter, another one is enhancer. In between this promoter and enhancer, we have different gene sequences. It will identify the protein coding exons, then it will mark the regulatory sequence by identifying the promoters and enhancers, then locate the introns and exons, then it will assign functional roles to sequences. So, it is very important in case of your human genome project and enables functional genomics. And in comparative genomics, we have goals like determine genomic structure and function relationship between species. We have different species like E. coli, Escherichia coli, bacteria, that is your plant, then worm, then human being. You can mark the base pair or genome size in E. coli is 4.6 million. Then plant is 125 million, but in human being it is 3 billion. So, under this 3 billion we have 20,000 to 25,000 genes. So, we have 3 billion base pairs and approximately 20,000 to 25,000 genes in human being. So, from this genomics we can compare different genomics of different species, and we can we try to understand how these will helpful in identifying different behaviors or different changes, different disease, different physiological changes in human being. Then synteny analysis is conserved gene order across species. Ortholog identification is same gene in different species. Phylogenetic reconstruction, it means evolutionary relationship. Mutation tracking is point mutations and chromosomal rearrangements. Now, bioinformatics in drug discovery, as we have seen, it is one application. Traditional approach, what we do, limited drug targets, trial and error screening in a human being. We don't know which dose will appropriate for a particular person. So, in the trial and error basis, we try to solve the or suppress that disease. That is why high failure rate are there, and it will take a longer time, and it may have some non-specific side effects. But in case of your bioinformatics approach, let's say we have 500 plus identified targets in case of your human genome. Then we go for computational drug design, that for a particular disease what should be the composition inside one drug. Then rational target selection, a particular target will be selected, it is not as a holistic manner. Then molecular docking simulations. Instead of applying something, we can simulate through models and software tools and algorithms. Then a personalized medicine will be developed, because if from the database of a person, if it's a sequence, all these genomics is known, all the sequences, then we can develop algorithms to find out the doses, to find out the composition of a particular medicine for that particular person. So, more effective than targeted drug with fewer side effects, because we are designing for a particular problem. We are developing that drug for a particular problem, not as a holistic manner or in generalized form. Then personalized medicine and pharmacogenomics. Pharmacogenomics means study of how genetic variant affect drug response. Time to time, we can mark drug will be resistant, the body will be resistant to drug. So, it depends on the genetics variants. So, problem is that different patients respond differently to same drug depending upon their genetic. So, this genetic polymorphism causes drug metabolism variation. Some drug fail due to adverse side effects in subset of patients. So, what should be the solution? Solution lies in genetic testing before treatment. So, if we know the sequence of the patient DNA variants, then we can predict the drug metabolism efficiency. Nowadays, people are using AI/ML for the prediction of this drug metabolism in the body. Then select the optimal drug and doses. Once we model that, once we know the sequence of DNA variant of a particular person, we can simulate that, we can check and we can predict how this effectively or efficiently the product drug will metabolism efficiently. So, then the select optimal drug and doses can be given. It will reduce the adverse effects like side effects and treatment failures, because we are not going for a trial error method. We first simulate, we first check it, we are first genetically testing it using some computational tools, then we are deciding which drug to be given and what should be the dose, what should be the composition. So, clinical impact will be like right drug, right dose, right patient. So, this is the optimal outcome and the biggest advantage of personalized medicine, which is coming under your bioinformatics. Now, understanding the disease at molecular level, basically the genetic basis of disease. So, every disease has genetic component like inherited or acquired. Either the disease has been inherited from their parents, direct genetic mutation will occur, so that due to this genetic mutation, disorder will come in the body. And acquired means due to environmental stress, genome will alter. So, that will cause like your cancer. So, for example, disease like cancer, genetic basis is oncogene mutation, and application can be your target drug development. So, as it is an acquired disease or genetic component, so target drug development is the can be possible by understanding this mutation at molecular level. Similarly, diabetes, that is through metabolic pathway disruption. This metabolic pathway, it can also be detected beforehand by understanding the at the molecular level. So, if we know, let some person previous generation of a person, the database is known that all these persons, they have all the previous generation, they don't have this diabetic, they are not diabetic, but in this generation the first time the person is diabetic. So why? It can be understood by looking behind that mm, genetic components, and from that it can be detected what will be the biomarker for this particular problem or for this particular diabetic person. So this can be, means through that biomarker can be detected. Similarly, hemophilia, it is also gene replacement can be done and the problem will be solved. So at molecular level we can diagnose, we can detect, and we can also find the solution. So it will give rise to better diagnosis method and targeted therapeutics and preventive strategy. If some person knows that beforehand, he knows that at certain age I will going to get diabetes or a particular disease, chronic disease, then he or she must take the precautions, and after certain that at that age he will never get that disease. So agricultural and environmental applications also there, like crop environment, identifying the genes for desirable traits, drought resistance, selecting the genetic and breeding, then pest resistance, nutritional enhancement, yield optimization, everything can be adjusted by adjusting the genotype. Environmental bioremediation, climate studies from microbial response, alternative energy from biofuel development, antibiotic resistant tracking from monitor resistance genes, forensic is DNA fingerprinting for support suspect identification, and microbial signature for source tracking. Multiple number of applications are there. Then protein structure prediction, it is the one of the most important in bioinformatics, usefulness of bioinformatics. And here the challenge, easy to determine primary structure but hard to predict the three-dimensional structure of protein, because we don't know how many different types of folds are there in a protein, and it is possible a numerous number of multiple folds, a numerous number of folds can be possible for any structure, and so many structures can be imagined or can be possible. So previously what we are doing, previously through experiments people are trying to find out or determine the protein structures through X-ray crystallography, NMR spectroscopy and cryo-EM. But gradually, as the development of this bioinformatics, people are trying to develop the software tools which has the computational power to model different model and predict different protein structures. So homology modeling, ab initio prediction through machine learning, AlphaFold nowadays, last year AlphaFold is given much more importance. So level of structure, primary amino acid sequence, then in secondary alpha helix and beta acids, in tertiary full 3D folding and multi subunit complexes. So because these are the different structural labels in different manner, primary, secondary, tertiary, like that. And why it matters? Structure determines functions and prediction accelerate drug discovery. So all are related. So in our life we have organism, the system organs, then cell is your phenotype, organelle is a structure, biomolecule is a function, and bioinformatics focuses on DNA, RNA and protein synthesis. Bioinformatics focus to understand the lowest level to predict all higher levels. means we are trying to operate in molecular level at DNA, RNA and protein level, and it will predict the higher levels and it can cure different diseases in the upper stages. Then cell cycle and gene expression control. We have different cell growth cycles, G1S, G2M, GO, and we have different signaling paths like complex chemical network allow cell to synthesize new material, break down waste for spare parts and receive signal to grow, divide or die. This is the signaling pathway. And the gene expression controls DNA to RNA transcription, that is a regulation which gene turns on or off. From RNA to protein, it is translation regulation which proteins are made up of, and post-translational protein modifications, and epigenetics is your chromatin modifications. The Human Genome Project. These are the milestones achieved in 2003. Complete sequence of human genome of 3 billion base pairs are mapped. 22,000 to 25,000 genes are identified. 17 other species subsequently sequenced, and this will revolutionize the impact on personalized medicine that is possible now. Then disease mechanism is unveiled, drug target identified, about 50 plus viable, and evolutionary relationship are clarified. Now some omics revolution means genomics, study of complete gene, DNA sequence, gene and chromosomes. Transcriptomics, study of all expressed RNA, gene expression pattern and regulation. Proteomics, which means study of all proteins, locations, protein structures, their functions and interactions, and it will give rise to the identification of all protein in pathway, cell and tissue. They will give rise to quantification of protein amounts and detection of modification and interactions. Then metabolomics is the study of small molecules and metabolic pathways and biomarkers. Then phenomics is the study of organism phenotype, genotype, phenotype correlation. All these combiningly give rise to an idea or understanding of biological processes. Then some computational methods in bioinformatics. Some are sequence analysis algorithm like dynamic programming, BLAST is for rapid similarity search, hidden Markov models for protein family detection, phylogenetics is for evolutionary tree construction, and in machine learning approaches are nowadays very popular. Previously neural network are popular, so that is AlphaFold for structure prediction, random forest model, support vector machine model is useful for protein function prediction, clustering is useful for gene expression grouping. And finally system biology, it includes network analysis, that is your pathway topology, metabolic modeling, cellular behavior prediction and simulation that will give rise to emergent properties. What are the current challenges or future direction of this bioinformatics? Current challenge is data volume, exponential growth, storage and analysis demands. It will be one challenge for bioinformatics field. Then data quality, in some cases they are incomplete annotations and error in database. Interpretation, converting data to biological meaning, integration of linking diverse omics data types, translation moving from lab to clinic application, ethics, privacy, genetics, discrimination, bioweapon potential, all these things we need to take care, otherwise these are the challenges. So future directions includes advanced AI/ML for structure and function prediction, real-time genomic analysis before any personalized medicine, prescription, synthetic biology design, single cell bioinformatics, microbiome and environmental applications, precision medicine at a scale. So, these are the useful and very useful application areas or future directions of bioinformatics. So, finally, we've got one summary of this. Bioinformatics includes biology, computing and statistics, and we can achieve the sequence entire human genome through this bioinformatics. We can identify gene disease relationship, how to develop the personalized medicine by understanding the gene disease relationship, and we can go for accelerated drug discovery and revolutionize the biotechnology area field. So, three sub-disciplines are like sequence, then structure, then function. Bioinformatics remain in infancy. Now, this is the started with unlimited potential for understanding and treating diseases. Thank you.